Hello students, this is Dr. Faizan Mirza. We are discussing energy and respiration. Why do we need energy? Energy is required for various functions in the body. For example, anabolic reactions. Anabolic reactions are reactions where we synthesize large molecules such as lipids, polysaccharide, proteins, and nucleic acids. We need energy for those processes. Similarly, active transport is a process where we need energy because sodium-potassium pump, for example, on our cell membrane, it uses ATP to maintain the ionic gradient and the resting membrane potentials of the cell. Mechanical work, muscle contraction, the movement of cilia, the movement of flagella, or intracytoplasmic movement, they all require the energy from ATP. Organisms which are ectotherms or organisms which are endotherms, we know that they are cold-blooded and warm-blooded respectively. Warm-blooded organisms maintain body temperature in a narrow range via negative feedback, and for that ATP is to be used. For example, in muscle contraction or in the brown adipose tissue, the energy is used up to maintain body temperature, at times to retain heat, at times to shed off excess heat from the body. Ectotherm, on the other hand, are cold-blooded. They cannot maintain their body temperature. Their body temperature is the same as their environment. How is ATP produced? How is energy produced? We use glucose molecule, which is C6H12O6, to combine with oxygen. 6 oxygen molecules combined with it to be converted into 6 carbon dioxide molecules and 6 water molecules, plus 2870 kilojoules of energy. So basically, we need energy for work done. And all these are just the name examples of why ATP is required and what's the use of ATP in a living organism. We can probably just think of some more use of ATP apart from these as well. When ATP is produced, we know that glucose is a hexose sugar, a 6-carbon monosaccharide, which is commonly respired, and ATP is produced from it. So C6H12O6 is a very stable compound. It would not react on its own. So how do you make it react? You make it react by providing energy to this compound, the activation energy. So energy must be added to make glucose react, and the energy is added either by providing the energy through phosphorylation or through using enzymes. So these two processes are used to provide the energy to glucose. So glucose on the energy profile diagram is somewhere here. Substrate combining with oxygen. You need to provide activation energy. You add ATP, so it becomes glucose. You add actually 2 ATPs to glucose, so it becomes glucose bisphosphate. And the activation energy is provided because of these 2 phosphates which are added to glucose one after the other. Glucose becomes unstable and then it starts to react. And the final products are carbon dioxide and water. And this is the amount of energy available in a glucose molecule. We can just compare that the reaction of glucose undergoing oxidation is pretty similar to burning a spoonful of glucose in oxygen. In this case, the energy is released right away at the same time, and it is all lost as heat because the reactant glucose and oxygen turns into carbon dioxide and water, and all the energy is lost as heat. But when the same reaction is occurring in our muscles, the reaction occurs over time. Gradually, step by step, glucose is broken down, and in these steps, there are various small steps here where ATP is produced. So the gradual release of energy is successfully converted to work done and ATP is produced. ATP is an energy currency molecule. An energy currency molecule refers to a molecule which is an immediate donor of energy. What makes ATP a suitable energy currency molecule? It's a small molecule. It can easily be hydrolyzed. It can be transported all across the cell. Yes, it cannot cross the cell membrane though. It's a water-soluble compound, a water-soluble molecule in fact. So adenosine triphosphate stores energy on these bonds, the phosphate bonds, and you break these phosphate bonds to release this energy. For example, hydrolysis of ATP can be taken into consideration that ATP combined with water to form ADP, and an organic phosphate is removed and 30.5 kilojoule energy is released. This is what we do whenever we want energy. We break ATP into ADP and PI, and we get 30.5 kilojoule of energy from it. Further, hydrolysis of ADP provides us with AMP, another 30.5 kilojoule of energy is released as another inorganic phosphate was removed. But addition of another, the hydrolysis of AMP further produces adenosine and less energy is released, this time just 14.2 kilojoule energy. Why the last bond releases less energy? Because the energy stays trapped in adenosine, because adenosine retains the energy to stay stable. Plus, all these bonds, the bonds which were initially broken are the phosphate-phosphate bond, and this bond is between phosphate and adenosine. The nature of bond is different, so its bond energy is different as well. ATP is an energy currency molecule, but there are energy storage molecules as well. Energy storage molecules are those molecules that store chemical potential energy. For example, either for short-term basis or for long-term basis. Short-term energy storage molecules include glucose and sucrose, and long-term energy storage molecules include glycogen, starch, and triglycerides. This is the reaction that we carry out for breaking ATP and releasing energy. We do not convert ATP into all these compounds. We just use this reaction to produce energy, to provide us with energy. And if we need more energy, we break more ATP molecules. We need more energy and we break yet another even more molecules. We don't just keep on converting one ATP into all these compounds, because energetically this reaction is feasible because once nutrients are available and oxidation is occurring of glucose, so this reaction can reverse and ATP can be produced back. The structure of ATP can be recalled that the ATP is having a pentose sugar, and you can recall whether it was a ribose sugar or deoxyribose. We know that the deoxyribose sugar is not there. It's always having a ribose sugar. So ribose sugar is there. There are 3 phosphates and an adenine, and the energy is stored on these bonds. When ATP is produced, how ATP synthesis occurs? ATP synthesis actually occurs by the process of, there are 2 processes by which ATP synthesis can occur in a cell. Substrate level phosphorylation or through oxidative phosphorylation. There are 2 types of phosphorylation that produce ATP. What is substrate level phosphorylation? Substrate level phosphorylation refers to how an enzyme is involved in producing ATP. Here, the enzyme will hold a substrate, which the substrate will already be having an inorganic phosphate attached to it. The same active site will hold ADP, adenosine diphosphate as well. The reaction will be catalyzed by this enzyme, and phosphate group from the substrate will be broken and will be added to ADP to make ATP. The substrate will be converted to product. You can see that the shape of the product is different as compared to the substrate. And ADP, when it combined with inorganic phosphate, its shape will differ as well because it turns into ATP. So the products, they leave the active site, and we see that ATP is produced and the product is released as well. This is called as substrate level phosphorylation. ATP is also produced using oxidative phosphorylation. Oxidative phosphorylation sums up 2 processes: electron transport chain and chemiosmosis. We'll be discussing electron transport chain later. This membrane here is the membrane of mitochondria, the inner membrane of mitochondria. This protein, the membrane protein, is called ATP synthase. This ATP synthase is an enzyme that can add ADP and PI together to form ATP. But this reaction works only when H plus ion cross through this protein channel. So ATP synthase is an enzyme that produces ATP by combining ATP and PI, and it's a protein channel as well. This protein channel allows H plus ions to move from a region of high concentration to a region of low concentration by a process of facilitated diffusion. This is called as the proton motive force. As H plus move from a region of high concentration to a region of low concentration, the energy is released, and this energy is used to couple ADP and PI together to form ATP. This process is called as chemiosmosis. The regions here are within the mitochondria. So this is the intermembrane space. This is the intermembrane space of the mitochondria. This is the inner membrane of the mitochondria, and this is the matrix. So when chemiosmosis is occurring, H plus ions start to move from the intermembrane space to the matrix. We know that this phospholipid bilayer is essentially impermeable for H plus ions. The H plus ions being highly polar, they cannot cross the phospholipid bilayer because the non-polarities would not allow that. The only pathway that H plus ion can move down the gradient is through this channel, which is ATP synthase. Later on, we discuss that oxidative phosphorylation is actually a step in aerobic respiration where ATP is produced. And substrate level phosphorylation is the process by which ATPs are produced in the stages of glycolysis and Krebs cycle. So when ATP synthesis occurs, there are two processes of producing ATP, as already discussed. We need to couple ADP and PI together to form ATP. One of the ways of ATP production is substrate level phosphorylation, as already described diagrammatically on the previous slide. The other is oxidative phosphorylation, which I'll be discussing on the next slide. What happens in substrate level phosphorylation? There's the addition of an inorganic phosphate to a compound. This does not need any proton motive force. External electron carriers are not required here. They are not needed. And the rearrangement of bonds release enough energy for ADP and PI to combine together to form ATP. This is substrate level phosphorylation. Substrate level phosphorylation occurs in cytoplasm of a cell as well as inside the mitochondrial matrix. In cytoplasm, when substrate level phosphorylation is occurring for one glucose molecule, 4 ATPs can be produced. 2 ATPs are used up, so there is a net gain of 2 ATPs. This reaction in cytoplasm can occur in aerobic as well as anaerobic conditions for one glucose molecule. So, a glucose molecule, when present in cytoplasm, can release 4 ATPs, 2 of which get used up, and they can this reaction goes on in absence of oxygen as well as presence of oxygen. What about the mitochondrial matrix? When substrate level phosphorylation occurs in mitochondrial matrix, 2 ATPs are made, and that's by aerobic respiration only. So for this substrate level phosphorylation to occur in the matrix of mitochondria, oxygen is mandatory. So these 2 ATPs for the same glucose molecules cannot be made if oxygen is not available. What is oxidative phosphorylation? An ATP synthase occurring through oxidative phosphorylation actually refers to how this differs from substrate level phosphorylation. Basically, it couples oxidation and phosphorylation through electron transport chain and chemiosmosis. This needs a proton motive force, as already discussed by chemiosmosis. External electron carriers are required where the electron transport chain will occur. And this is occurring only in aerobic conditions. Oxidative phosphorylation occurs inside the mitochondria and occurs on cristae along the inner membrane of mitochondria. One glucose molecule ends up producing 28 ATPs. So you can see an immense increase in the ATP production capability of one glucose molecule when it undergoes oxidative phosphorylation. We also discussed coenzymes in relation to respiration. There are two types of coenzymes relevant for us, hydrogen carriers and coenzyme A. Hydrogen carriers are of two types, NAD and FAD, that we'll be discussing. NAD stands for nicotine amide adenine dinucleotide, and FAD stands for flavin adenine dinucleotide. They both can carry hydrogens. They carry 2 hydrogens each, and the hydrogens are in ionic condition. So when hydrogen is carried by NAD or FAD, it forms reduced NAD or reduced FAD. Coenzyme A. Coenzyme A is another coenzyme. This is a carrier of acetal. Acetal is a 2-carbon unit. And NAD and FAD, as already discussed, they are the carriers of hydrogen. That's it from my side. Thank you so much.